Welcome to this explainer. Today, we're taking the incredibly complex world of cell division and turning it into a clear, highly visual master plan. If you've ever found yourself staring at textbook diagrams and just feeling totally lost, well, you are in the right place. We're going to break down the exact biological mechanisms you absolutely need to know to completely master your O-level biology exams. So, let's get into it.
Think about that for a second. Literally millions of new cells are being created right now, just as you're sitting here watching this. But how exactly does that whole process work? To answer that, we're going to bridge the gap between the macro effect, the stuff you can actually see, like healing a paper cut or growing taller, and we're going to zoom straight into the microscopic cause to look at the invisible machinery that's constantly keeping you alive.
Here is our roadmap for today. First, the body's master plan. Second, anatomy of a chromosome. Third, the cell cycle explained. Fourth, mitosis, the perfect photocopy. Fifth, meiosis and genetic diversity. And finally, some crucial exam tips and a recap.
Okay, Section 1, the body's master plan, focusing on growth and repair.
Notice this macro view we're showing here? It's just a simple scratch on the skin healing over time. But then look at the micro view alongside it, showing the underlying cellular action. The body's ultimate master plan is essentially creating millions of new cells for growth and repair, all stemming from just a single cell. Understanding this invisible machinery is absolutely crucial for seeing exactly how one single starting cell can develop into a highly complex, fully functioning organism like you.
Moving right along to Section 2, anatomy of a chromosome, the molecular blueprint.
Imagine for a moment that we're shrinking all the way down into a cell's nucleus. Right here, we find these incredibly tightly coiled packages of DNA. These are your chromosomes. They act as the absolute instruction manual for life, carrying all of your genetic information in the form of genes. Just take a look at how densely packed this DNA has to be just to fit inside a microscopic nucleus. It's kind of mind-blowing.
Now, before a cell can divide, it has to double its information, right? Notice how the single chromosome replicates. Suddenly, we have two perfectly identical copies, and we call these sister chromatids. This step guarantees that each new cell is going to receive the exact same genetic information. And check out that physical joining point right in the center, clearly labeled there. That is called the centromere. It's exactly what holds those two identical blueprints together before they're finally pulled apart.
So, it's super important to contrast two concepts that students constantly confuse. Over on one side, our sister chromatids are exactly identical copies, but on the other, we have homologous chromosomes. In sexually reproducing organisms, you inherit one of these from each parent. They're similar in size, they have genes in the same locations, but those different colored bands you see, those represent alleles, which are basically just different versions of the same gene. This specific setup is the entire foundation of genetic diversity.
Let's hit Section 3, the cell cycle explained, taking a look at a step-by-step flow.
Following this chronological timeline, you can clearly track how a cell actually spends its life. Step 1 is interphase. This is essentially the big prep phase where the cell grows and replicates its DNA, creating those sister chromatids we were just talking about. Once all that prep is done, the flow moves straight into the M phase, which is mitosis, and that's followed by cytokinesis, which is where the cell finally, physically, splits into two.
Which brings us to Section 4, mitosis, the perfect photocopy in somatic cell division.
The best way to think of mitosis is like a biological perfect photocopy machine. It operates exclusively in your somatic cells, so your basic body cells like skin and muscle. Its entire purpose is growth, repair, and asexual reproduction. You have one parent cell go in and exactly two genetically identical daughter cells come out. And the most critical data point to remember here, the chromosome count remains exactly the same as the parent cell. Absolutely no information is lost.
On to Section 5, meiosis and genetic diversity, specifically germline cell division.
Now, unlike the photocopy function of mitosis we just saw, meiosis occurs strictly in germline cells. Notice the specific locations highlighted here. It takes place exclusively inside reproductive organs, specifically the testes and ovaries. The sole purpose of this pathway is to produce gametes, sperm and egg cells, for sexual reproduction. It fundamentally cuts the chromosome number in half, so that when two gametes eventually combine, the resulting organism has the exact correct amount of DNA.
And this next part brilliantly illustrates exactly how you get your unique traits. If we magnify a very specific stage of meiosis called prophase 1, we see this phenomenal molecular event. Notice the physical exchange of DNA segments between the non-sister chromatids. We call this crossing over. See that glowing X shape where the structural arms intersect? That specific crossing point is called a chiasma. This precise mechanical swap is literally what greatly increases genetic variation. It's what makes you entirely unique.
Finally, Section 6, exam tips and a recap to help you secure those marks.
Let's look at a very frequent error made on exams. Examiners absolutely love to test if you know the difference between these two terms. So stamp this side-by-side comparison right into your memory. Homologous chromosomes have similar size, similar genes, but different alleles, so they are not identical. Meanwhile, sister chromatids are attached at the centromere, and they are perfect, exact, identical copies resulting from DNA replication. Do not mix those up.
Okay, let's rapidly recap to consolidate all of this. 1. Chromosomes are tightly coiled DNA packages. 2. Homologous chromosomes come in pairs, one from each parent. 3. During prophase 1, crossing over at the chiasma massively increases genetic diversity. 4. Mitosis is that perfect photocopy machine for somatic growth and repair. And 5. Meiosis is the specialized division in germline cells specifically for sexual reproduction.
Now for a major exam tip. Think about this really common question. Complete the diagram to show how a parent cell produces two daughter cells. The absolute best way to guarantee you don't lose silly marks is to literally practice drawing out these components and stages yourself on a piece of blank paper. Make sure you can actually draw the parent cell maintaining its chromosome number in mitosis versus cutting it in half for meiosis. Physically label your centromeres, label your chiasma. Muscle memory totally counts here. As we wrap up this explainer, I'll leave you with this final thought. Right at this very moment, there are millions of microscopic, flawless, cellular photocopies happening inside your own body, running this incredibly complex flowchart of life. It's an absolute marvel of biological engineering, really. Keep reviewing these pathways, keep practicing those diagrams, and you are going to walk into your biology exam with total confidence. Thanks so much for watching, and keep exploring the incredible mechanics of life.